So today we're going to be talking about endocrine control of exaggerated trait growth in rhinoceros beetles. So it has been studied and shown that juvenile hormone and insulin signaling pathways do lead and contribute to the growth of horns and weapons in many forms of insects. Specific research into stag and dung beetles have shown that juvenile hormone and the insulin signaling pathways are specifically the reasons that these insects grow these exaggerated horns. And research into them has shown that juvenile hormone is just one of the large reasons that they grow these horns. So the purpose of this particular study is to explore how the juvenile hormone affects the development of weaponry. So for this particular study, the question that they're focusing on is how does this juvenile hormone affect the growth of weapons in T-dichotomous beetles or Asian rhinoceros beetles? They hypothesized in the study that along with the insulin signaling pathways that juvenile hormone would be responsible for growth of the horns in these beetles. They hypothesized specifically that the larger males would have a higher level of juvenile hormone, and that when they apply the juvenile hormone, that larger males will result from the larvae that had the juvenile hormone applied to them. They also believed that these larvae that had the juvenile hormone applied to them would also have larger horns as a result of the juvenile hormone. So now I'm going to pass it on to Hannah to talk about the methods.
All right, so to begin, 2 experiments were actually run. There was a control experiment and a treated experiment. So the specimens that were in the control experiment were given a treatment of acetone as sort of a placebo, and the treated specimens were actually given phenoxycarb, which is a juvenile growth hormone. And right here I was showing the different stages: larval, pupal, adult, since we are focusing primarily on the larval and pupal stages. All right, so right here I was going to show you the phenoxycarb and acetone structures. All right, so the phenoxycarb and acetone were actually applied behind the head ectopically, and they applied it in 3 different stages. So they had a group that had the phenoxycarb applied at the late prepupal period, which is shown right here actually, and this is when the most horn growth occurs. So this is sort of like the most important group to really observe. This is the larval feeding period, which is at the end of the 3rd instar. This is the early prepupal period, and both of those also had test groups that had phenoxycarb or acetone applied. All right, so the larvae were all initially sampled. They used an X-Acto knife to make an excision and a pipette to actually extract the samples. And after the initial sampling, 20 males were divided into 2 different groups. The first group was actually resampled at the early prepupal period, and the second group was sampled again at the late prepupal period. And the samples from the control group actually showed that the JH titers were the highest during the late feeding period. All right, so the larvae were all purchased from the same Japanese retailer to eliminate sort of like variation in the way they were hatched and reared up until the scientists acquired them. The larvae, after they were acquired by the scientists, were all housed in identical environments, so moist soil, hardwood, and all the larvae were provided with all the food they could eat. This is to eliminate any variation that might have been caused by having different diets for each of the larvae. All right, now I'll turn it over to Christian for the results.
All right, so we're going to be talking about the big ideas and the outcomes of the experiment. So here's a complete collection of all the data they got in a table, but these are a little congested, so we're going to take a look at it in the form of graphs. So the first big outcome was that the JH titers did decrease over the course of larval development. As you can see over here in the late feeding period, the mean JH titer concentration was higher than over here in the early prepupal and late prepupal, from which they basically were about the same as each other. The JH titers did correlate with body size throughout development. Over here it shows you the JH titer versus the larval weight on the x-axis. So you can see as the JH titers went up, the larval weight also went up, and that was the trend. at least in the larval and early prepupal stages. During the late prepupal stage, regardless of if they were small or large, their JH titers were relatively uniform. And for the final big outcome, the JH analog did not really affect horn growth. This was the results of the experiment. They have one line, the black bold line, which is representing the acetone control group, and then the dotted line is the JH. So you can see that for the majority of this, they're relatively in line. So it was just about the same results for each, except for over here where there's a negligible difference. And here we have the results of the horned beetles. One was treated, this one right here was treated, this one was not, as opposed to the same experiment that was conducted on stag beetles, where this one was untreated, but this one was, and it got the exaggerated trait growth of the weapons. So now I'll turn it over to Clay for the discussion.
Weapons, the main topic of the study. Found in different shapes and sizes and mainly used for male competition. This experiment had negative results, but even with negative results in research, a negative can still be a positive. With negative results, you still have progress towards the research, and there's a next step that you can take. The next step in this experiment, they wanted to try to see if juvenile hormone was redundant to the outcome of insulin signaling pathways. So they wanted to block the insulin signaling pathways by using double-stranded RNA to actually knock down the signaling pathways. And they wanted to see that if the juvenile hormone could actually save the horn size, even though the insulin signaling pathways were no longer helping towards the growth. Are there any questions?
Woo!